This is where we left off. We just barely got started on this particular slide. And a couple of things I want to add to what I was saying before is these two chromatids, these sister chromatids, are attached to one another by a chemical called a cohesion. We aren't actually defining what that chemical is, but the sister chromatids are, it's kind of a cohesion or a stickiness, plus you actually have the centromere is an area where there are proteins that are even more attached. As we move on with this process, you understand that that centromere plays a role with regard to the attachment of microtubules that are actually going to attach and be able to pull these two sister chromatids apart, but they're actually attached to each other all along the length of these sister chromatids. And what you have with regard to that centromere, it's a narrow area, the centromere, the narrow area, and very much of a specialized region with regard to the DNA that you have there, that the above the that each of the areas above and below the centromere are called arms, are called the arms or pieces, and arms are what they're called, so I'll just use that term with regard to it. But what's going to happen is you're going to attach a microtubule to the centromere, and those microtubules are going to shorten and to allow these two sister chromatids to separate. Once they separate, then they're called chromosomes. This becomes a, I guess maybe a mystery or a bit of a riddle. Together, they're considered sister chromatids. When they separate, then they're considered chromosomes. And when they actually separate and they are attached to a pair, so they're attached to their pair that in soma, their duplicate number in soma cells, they're also called chromosomes. One of the reasons why we consider them sister chromatids here is they're actually mirror images of each other. They're exact duplicates. And so we're trying to make that kind of distinction between something that is part of the genome and then something that's a duplicate of the genome. In eukaryotic cells, the division of a soma cell consists of mitosis. And with regard to mitosis, that is the sum total of the division of the genetic material. And then after mitosis, you have cytokinesis, which is the division of the cytoplasm. So many times the phases are all put together, but actually you have this separate ideas that mitosis is just genetic material division, replication and division, and then all of the organelles, so cytokinesis, all of the organelles, the Golgi, the rough endoplasmic reticulum, all of that also has to be replicated, and that's cytokinesis. Gametes are produced, and here's that word gamete, which I've used before. That's the sex cells. Gametes, we call them sex cells, whether they're male or female, but what they have is a single set of chromosomes, and so they're produced by another type of division called meiosis, and this is where I had in the past been calling it meiosis, just so you can see what that word looks like, but it is meiosis the way it's pronounced. And this yields non-identical daughter cells that only have one set of chromosomes, so half as many as the parent cell. And a lot of this stuff is important. It seems like it's kind of just a fact, but it is a very important fact, particularly for those of you who are wanting to do very well on the test. It tends to be, you know, something that would end up in a matching or a multiple choice question. And so looking at this, and we'll bring this image back multiple times because it's a nice compare and contrast image. You do start out with learning and what the phases of mitosis is: prophase, metaphase, anaphase, and telophase. And then after telophase, you have cytokinesis. What happens in meiosis, the one that creates gametes, so single chromosomes, a single set of chromosomes, is we need to take a precursor cell, so we need to take the complement of a body cell and reduce it down into 23 individual chromosomes. And so what you have is that process of prophase, metaphase, anaphase, and telophase basically happening twice. It happens twice to actually reduce the chromosomal numbers to only 23 individual chromosomes. And with regard to the cell cycle, it's pretty universally decided that this is what the image is going to be to depict the cell cycle. It's very similar to the one that we had in the very beginning. And what you have is a complete circle. It is a cycle. And most of that circle, so about 90% of that circle can be defined as something called interphase. Only about 10% is this mitosis, is this mitotic phase, where you have the results of the end of interphase, then then separating the already replicated DNA into two cells. So let's go through these individual phases. And your book talks about on average, each cell is a little bit different. As a matter of fact, you have some cells in your body that are replicating every day, and you have some cells in your body that don't seem to replicate at all. Think of nerve cells. They replicate very slowly or not at all. And so when you're referring to times, or you're referring to, when your book refers to times of each of these phases, they're talking about on average. But if you start out on the left, you have G1. That's the first phase, or the first gap. And many times in this particular phase, the cell is just doing its cell stuff, like making proteins and using glucose to make ATP, etc. And then you have the S phase, which is given that name S for synthesis. This is where the DNA is replicated. And then the G2 phase is the second gap. So the DNA has been synthesized, and now it actually, there is a reason why you have these gaps, because you have in between the two, the G1 and the synthesis, and the synthesis and the G2, you have certain stops, or you have there are places in this cycle where the cell is actually monitoring whether the cycle is going well or not. And then you have the mitotic phase, which is made up of mitosis and cytokinesis. So if we're talking about the mitotic phase, we're putting those two parts of the mitotic phase together, and that's the last part of the circle. As I said, conventionally we divide mitosis into 5 phases, and here you see the words. Prophase, and basically in prophase, remember the chromosomes have already been replicated. What's happening in prophase is the chromosomes are condensing, and the nuclear envelope breaks apart. We have spindle fibers forming. Talk about more about the spindle fibers, but what happens is, remember, this single cell is getting ready to turn into 2. So if you remember back into the material we learned for the first test, just inside the cell membrane you have these microtubules that create the cytoskeleton. Well, those microtubules that are creating the cytoskeleton are kind of units that can be disassembled and assembled. And they're disassembled into these spindle fibers. So these spindle fibers form basically from disassembled microtubules of the cytoskeleton to create a arrangement that will actually be able to pull these chromosomes apart. That's prophase. And then after prophase, so the spindle fibers are forming to attach to each of these individual chromosomes. Metaphase, the chromosomes are forced to line up. They don't line up. What happens is you have spindle fibers attached to each side of the pair, and they're basically pulling each other. They're equally matched, so that makes those chromosomes line up down the middle. And then one of the reasons why I wanted to say that very, very tightly or closely about the cohesions between the sister chromatids is now an enzyme comes in that actually breaks that cohesion apart. So you have that cohesion no longer there between the sister chromatids and these spindle fibers pulling the chromosomes apart, and that's what happens in anaphase. In anaphase, the centromeres divide, chromatids move toward opposite poles. Remember, now they're actually considered individual chromosomes because they all both have the ability to replicate themselves. And then in telophase, the chromosomes uncoil, so they become less condensed, new nuclear envelope forms, and spindle fibers disappear. And you can see that there's also this idea of a cleavage furrow, this bending in, and then cytokinesis, the cytoplasm of the cell is cleaved and halved. Cytoplasm halved, organelles then are remade. Those are the 5 phases, and a little bit about what happens in each. With regard to this mitotic spindle, I don't believe I mentioned the centriole pairs. So you have a centrosome with centriole pairs, and the spindle made up of microtubules. And what it does is, and as I said, it's going to attach to the chromosomes and then pull them apart during this whole process between metaphase to anaphase. And the spindle microtubules are attached via the centrosome and then are pulled towards opposite poles of the cell, of the new forming cell. This centrosome starts out as one. It replicates during interphase, turning into 2 centrosomes that then migrate to opposite poles, opposite ends of the cell during prophase and prometaphase. So it also, this centrosome also has to replicate and move, and as it moves, these spindle fibers are created, and it's kind of like a fishing expedition. The spindle fibers actually throw themselves out to try to attach to the centromere of the chromosome. When they attach to the centromere of the chromosome, then they start pulling in their direction, but in the meantime a centrosome from the other pole is attached to the other side, and again this tug of war takes place. You have on here, on this image, you have at the top a dyed microscopic look at interphase, prophase. Remember, prophase is where that nuclear envelope is going to start disappearing. You're going to have these centrosomes move to the opposite poles of the cell and start attaching to these chromosomes. And then prometaphase, that process of attachment so that this tug of war can take place. And metaphase we actually think of as them lining up down the middle. The only other term that shows up here, there is a protein called a kinetochore, and that kinetochore becomes very important with regard to when we'll look at it closer to the spindle fibers. We'll pick this up on the next one.